Thank you everybody for joining. My name is Joel Orienti. I'm PhD in biochemistry at Nanome. I have Steve Baker here with me, and we're just going to do a quick intro together, and then we're going to hop into Nanome and give an example of what somebody might do for designing a new drug molecule, starting from the crystal structure with a docked compound. So yeah, Steve, take it away.
Hey Joe, yeah, Steve Baker. I have an undergraduate degree in medicinal chemistry and a DPhil in organic chemistry. Started my career as a medicinal chemist at Wellcome Research Laboratories back in the 90s. But then after my PhD and postdoc, went to work for Anacor Pharmaceuticals in California and became the head of medicinal chemistry there. During my tenure there, we got several molecules into the clinic and 2 NDA-approved compounds. From there, I went on to GSK, became the head of medicinal chemistry for the antibacterial unit and the infectious diseases group. And from there, went on to Janssen to become head of emerging immunotherapies in the immunology discovery group. And from there, went on to become the head of chemistry at Reactive Biosciences.
That's awesome. Sounds like a super fun career.
It's been good. Yeah, I've enjoyed it.
My experience with Nanome, I've really enjoyed this software. Just figuring out, being immersed in the protein itself and just looking at the space around the protein. I think one of the best features for me is being able to put the protein into a particular configuration, and then me moving around that protein, moving my head in and out, and just seeing it that way. Really gives me the it really immerses me into the protein and see things I haven't seen before, just using the molecular modeling on a 2D computer screen. So we'll show some of that.
It's amazing. It's amazing what you notice in a 3D environment compared to just what that static 2D flattening of a 3D object does.
Yeah. Just finding new residues, finding new interactions, looking at the way the ligand interacts with the protein. When you can just hold the protein steady and look around it yourself, just move around the virtual room, it's given a real new approach to molecular modeling for me, and giving me ability to design new compounds that I wouldn't have even thought about, I think, using the regular software. Okay, so let's load up a protein. This is PI3K-alpha, and so this is quite a popular protein target for oncology. And there's a couple of published structures with ligands, and so what I wanted to do was understand, how do those ligands bind to that protein compared to each other? So we'd heard that they were in similar within a similar space, but it was really kind of curious to me, how did how do these two ligands overlap? What are the key interacting features of those two ligands? And then is there a way of making like a composite ligand of both of those, taking into account both the best interacting groups, at least, that we can see by through the modeling? So that's what we've done here.
So these are the same protein scaffold, but with 2 different compounds in the same pocket.
So this is PI3K-alpha, and this is these 2 ligands here. You can see how they're overlapping, and we've created a structure here where we've got the residues within a 5 angstrom space of the 2 ligands. So, and these are binding to what's known as the helical allosteric site. So most of the protein you can kind of ignore, but I like it there just to remind myself it's the protein. And so now I can zoom in and just hold it there in space, and now I can have a look around to see how are those 2 ligands overlapping, and where are those interactions. So we can start taking some of these away. So if I just take away the ligands themselves, so now we're just looking at the protein, and you can see here it still has the interacting sites, so I can see where the interactions are coming from, but I can now have a look at the space fill in that area and see all the different residues around that. So let's bring one of these back. So the first one, so this model that we're looking at the moment, this is the 8-TSD PDB. So I'll bring up the 8-TSD ligand, and so that's the ligand that goes with this protein that's been published. And so we've highlighted that in this pinkish color to just make it more obvious over the rest of the molecule. And you can see there's some really key interactions here. You can see this aromatic ring in this plate, in this position here, has got a nice pi stacking with this tyrosine down here. There's a nitrogen over here that's pi stacking, I guess, with this phenylalanine. Hydrogen bonding over here from this nitrogen to this oxygen. Interestingly, not really seeing anything from this oxygen and this amide. No real hydrogen bonding there showing up, at least in this structure. Chloro group here interacting with the backbone carbonyl. And so quite a lot of good interactions going on here. Again, edge to face pi stacking again here with this molecule.
Yeah, I think it's really cool how we have both, like, you've got aromatic interactions, there's some hydrogen bonding, there's, you've got both aliphatic groups and polar, like this molecule's interacting with a lot of different types of chemical interactions from this protein.
It's been well optimized for sure. Yeah. You can see over here a trifluoromethyl group, also hydrogen bonding with a hydroxy group here of a threonine. So what's interesting with this molecule is it has kind of a V shape. It's got two different substitutions going off in two different directions, and then it's got a tail here. So it's kind of a Y-shaped overall type molecule. If you can see that. So that was the 8-TSD. So if we look at the other protein now, which is 8-TDU, published. So again, here's the structure of the protein, and let me pull up the ligand, make that visible. So now you can see the ligand. We've highlighted this one in green. This is more like a dog leg type structure. Let's just say it's. You can see it's a smaller overall molecule, but again, you can see how this is interacting. And you can see again pi stacking going on with this group here. Again, there's a fluoro molecule over this end of the molecule, hydrogen bonding to the hydroxy group of the threonine. This particular molecule, though, this has a urea function that's got some nice hydrogen bonding network going on in there, and again another edge to face type pi stacking going on in this part of the molecule. Hydrogen bonding between this nitrogen here. To this backbone carbonyl. So again, you can see a lot of effort has gone into the optimization of this molecule as well.
Yeah.
So I wanted to understand, when I saw that both structures come out, I wanted to understand how do these two structures overlap, and are they in the same site? Are they in different sites? Are they, how much of it is overlapping? And so doing this on a 2D structure is fine. It can, obviously molecular modelers can do that, no problem. As a medicinal chemist, for me, I just like this space, and I realized this is something I could do really quickly here. And so if I now open up the second ligand, we can now see how those two ligands are overlapping. And what I found really interesting was that it overlapped probably more than I was expecting. And the way the two different scaffolds interacted, or docked, should I say, in the protein, showed me that there is a way to try and combine both of these molecules into a single structure, and try and maintain all the best interactions from both sides. So as I mentioned before, let me just try and get this into a shape. Where did you go? Oh, you're over there. Okay.
Yeah, I moved.
So. Yep, that's okay. So you can see you can see the Y-shaped molecule of the pinkish structure. But what really struck me was the way the green structure kind of came straight down between the two of those substitutions coming off at the top there. But then the rest of the molecule,
branch, right? But it still takes up that space.
Yeah, it's come in at a different trajectory. So it is still interacting over in this space here that we saw before. It's still getting some pi stacking going on, and it's built that gap. It's built between the gap between those two branched units.
Yeah.
But then down on the stalk part of that Y structure, and on the dogleg structure of the green molecule, you can see now how these are overlapping pretty well, especially towards the end here. You can see they are taking up the same space as well. So the first thing that really kind of struck me was, that there was actually a lot more overlap than I was expecting to see. And And again, I've looked at this previously in 2D structures, but here it was really able to kind of immerse and have a look at that and see where the differences are. And then you can start seeing that there are some real key differences here. For instance, this nitrogen interaction here, this is interacting with the other structure that this was docked with, so you can't actually see that carbonyl group here, but we know that exists. But the green molecule isn't getting far enough to make that interaction. Right. So there may be an opportunity to build this type of structure onto that green structure to try and pick up more interactions there. I think again,
we still do have a carbonyl interaction here, which is pretty cool, right? They took a different approach of not filling up as much of the pocket, but still trying to grab some interaction.
They do. That is a key interaction for that molecule for sure. Just made me realize that there's more. This looking at the structure in Nanome though made me realize that there was more that could be done in this region with the green, where the green molecule kind of misses. There's a nitrile group coming off of here that's got a nice interaction, and a little bit close in this structure here, but again, I think it's still in the same region that the green molecule is missing. But then up the top of the other end of the molecule, you can see that the green structure here just looks to be a more efficient use of the ligand, so better ligand efficiency. It's not using two branching chains necessarily to try and create that same level of interaction there. So again, I haven't looked at all the SAR for all of these to understand exactly what's needed, what's not needed, and compared to the pink structure, how that was developed. But just looking at it in Nanome here, and as I can move around the molecule and move in, the green molecule to me at this part of this end of the molecule just seems to be a more ligand efficient molecule, whereas the pinkish purple structure down the other end seems to have a better chance of creating new interactions that seem to be missing at least with the green structure. So certainly an ability to create a composite. So as I was looking at this, I felt I'd probably want to keep that top end of the molecule with the green molecule. So we will keep that, bring that down, and you can see they start to overlap. Let me zoom in a little bit there. So I showed you the urea type structure on the green.
Right.
Interestingly, that starts to overlap with the amide on the pink.
Yeah.
So now we can use that as a point of connection. So we can bring the green structure down to this kind of point here. On the purple structure, there seem to be quite a few interactions up in this region here that the green isn't taking benefit of. So this to me became a good opportunity to take this green structure and start merging it with the pink structure here, and then perhaps
maybe add some more bulk up there.
Yeah. So rather than have this urea type structure, we could have like an aminopyrimidine group here, because this is the pink is an aromatic group. And then over here at the top of that, we could have an amide coming out of that. So that allows us to build a composite structure with an aromatic ring there. And once we put this aromatic ring onto the green structure, we can replace the green part down here with this purple part, and that just gives us the first step on a composite molecule of trying to incorporate both of those. The one aspect that is a little bit tight here, you can see, is this lysine. So the lysine has got this tight interaction here. Certainly with the amide, but it's clashing in this particular molecule. Yeah. And so as I was looking at that and realizing that, it's like, okay, it's got a lot of rotational bonds. It's got to be, we've got to be able to have some rotational freedom, some flexibility there. So perhaps that clash isn't happening. So as I was looking at the structure, I switched off the 8TDU structure, switched back to the 8TSD structure, and now you can see that the lysine here has moved up and out the way. So that is flexible. So it can go down. I'm sure we can maintain that interaction there.
It's not just rotational space of the R group from the lysine, right? This whole helix and loop has a lot of rotational freedom.
It has, yeah. And so So it made me realize, it might be worth in a composite molecule. Let's leave an amide group there, try and pick this up. There's a couple of lysines there that are going to give us opportunity to interact at some way. So to me, I'd like to keep the amide there for the moment and just see if that would add a potency to the composite molecule. The only other thing that I think that we would want to change to on the green molecule is you can see on the green molecule there's a methyl group here of that furan ring. And you can see on the pink molecule there's a chloro group that's interacting with this carbonar through a hydrogen bond. This methyl group can't do that. They're close enough. Let's just swap that methyl group for chloro as well and just see if we can pick that up. So keep the green, make the methyl a chloro, come through this urea group to this point here with an aminopyrimidine, keep an amide off of that, and then continue down here to the nitrile, and keep this terminal group of the pink molecule together. And so to me, then that became quite a straightforward composite molecule to design. Again, things that I could probably do in a 2D space. I just found it a lot quicker to review and analyze in the Nanome space.
Yeah, I agree. This is a fun little thought exercise of how do you combine two molecules, keep the best parts of each one, and be able to just switch back and forth from one protein scaffold to another and see, which physical space was taken up in one crystal structure compared to another, and then how do you optimize for both of those. But just in this 3D environment where nothing's flattened, it is just fully 3D.
And for me, I really like the idea of just being able to hold the protein in front of me and move my head around it. Because when I'm rotating it on the screen, I can lose. I'm a medicinal chemist, so I can lose things. I need a computational chemist to kind of highlight things for me. Whereas in this world, I just found as a medicinal chemist, being able to just hold the protein in front and just move my head through it, have a look at the various different interactions with the protein, just highlighted things I found quicker through in this virtual world.
Yeah, I agree. I really love the three-dimensional space exploration. It's a lot more intuitive. We have a pocket. That pocket is three-dimensional. Let's look at it in 3 dimensions and see what's built and where we can build into as well.
Yeah.
All right.
And
cool.
even just the use and speed.
Okay.
Being able to use it, just for me, just getting up to speed on the usability. It was a little bit of a learning curve, but it came really quickly. And being able to switch off and on the proteins, the ligands, being able to select the residues like we just did, it was a really quick learning curve, I found. So
yeah, it's cool. It's amazing how many people have some apprehensions on just hopping into the headset, but once they get in there, it's like a kid in a candy store of just they grab the protein, they make it really big, they get in the pocket, and they're just looking at residues, seeing which numbers they are, right, comparing two structures. And we've just watched a whole helix turn into a new conformation as well too. That's just pretty cool. All right, so let's hop over to Mara. Let's draw the new structure, and then let's use Mara to dock it for us, and then we can come back to Nanome and look at our products.
Dock it in. Sounds good. All right, see you on Mara.
All righty, so let's draw this molecule. Okay, so we wanted the green molecule for the left-hand side, so starting to draw that out. I'm going to start here with a benzene. Okay, and onto that we're going to add the fluoros, put the furan, get the furan in place, add the fluoros, make that an oxygen, make that the fluoro, and here we wanted this one to be the chloro, so we'll change that to chloro. Okay, that's looking right. Let's extend from there. Put the trifluoromethane in place, the right stereochemistry. There it is. Trifluoromethyl. Okay, now we wanted to put the wanted to start moving this to the other molecule and connect them through this amino pyrimidine group. So let's make that a pyrimidine. There we go. Put the nitrogens in place, and then we wanted to extend this out into the amide to try and pick up some interactions. And so we wanted that ortho to the top nitrogen. Okay, let's make that the amide. Put the oxygen in place. Nitrogen. Okay, and then we wanted to connect that to the pink purplish molecule. So coming off the ortho to that bottom nitrogen, we can then build the rest of that ring. Bonds in the right place. Okay, that's the molecule.
And so now we should be able to dock this at our target protein. We're going to generate a single conformer. All right, now that we have this single conformer from that smile string, we can use a docking protocol. So we have a workflow set up. We can open up our docking workflow. And in here, we want 8tsd.pdb, so we need to find the coordinates to dock at. That's our residue name. So we'll find the centroid of the ligand, and those will give us our coordinates for docking. We need to create a protein-only file from 8tsd.pdb. We'll add hydrogens. We need to take the output from step 2 and push it to step 3 so we can create a PDB-QT file for docking. And in our docking step, we are going to use the output from step 3, that PDB-QT file, as our receptor. We found the coordinates XYZ for docking in the centroid step, in step 1. We'll keep size 25 and exhaustiveness of 8. That's fine. Then we need to add our smiles SD file. All right, there we go. And then for visualization, we're going to combine the protein-only file from step 2 with the docking output from step 4. So we can go ahead and run that workflow, and Mara's just going to fire off and start going. So as the tools finish, we can expand them and see what our docking coordinates were. We've converted to a protein-only file. We can open that here. See that our ligand is gone. If any waters were there, they'd be gone as well too. We also created our PDB-QT file, which is going to just look like our protein-only file, but it's formatted for SMINA. And now our docking is running. We did this with a workflow that we had saved previously. You can look at our docking results here in 2 ways. This is the complex that we set up. So if we click on our ligand that we just docked, we can zoom in, we get a 5 angstrom sphere, and we can look at some chemical interactions. We can also go through the different poses and see how those chemical interactions will update. We can just, flip through all the poses here. We'll look at these more in depth in the virtual space in just a minute. But also, if we wanted to just look at our poses, the docking results are saved down here in the output from SMINA. So each individual pose exists here, and then we also have a docking scores of which we can see those. All right, so we'll move to Nanome now and look at our results in virtual space.
Okay, so here's the protein then. So the first ligand was the one in green. We can see that one now where that's located. And the second ligand that we were looking at Let me take that one out. The second ligand we were looking at is the one in pinky purple. And again, that's got the Y-shaped structure, so we're good there. Clashing with the lysine as we were looking at previously. So that's all good. So if we bring back both of the, and so if I take that ligand away and bring on the complex. Now this is the composite molecule of both the green and the pink, purpley pink. And so this is the first of the docked images. So let's have a look and see how well it went together with first the green molecule, because we were overlapping the green molecule with the composite molecule here. And that looks pretty good. There's a slight rotation to the benzofuran ring. The important fluoro groups are overlapping, as you can see back here.
That's pretty awesome.
That's really good. That's what we wanted. These fluoros are a bit further apart, so there's a bit of a rotation going on in there. If you remember, we wanted to make this methylochloro, so we can get an interaction up here. Let me just very quickly bring back the other ligand. So that was that one. So here you go. You can see the chloro group that we're trying to overlap. They're pretty close together, and I think that's perhaps what it was trying to do. So nice and together there. Let's just take the pink one away for the moment, purple one away.
Your CF3 in the back too is pretty nicely overlapped. At least meets in some position. The CF3 in the back here.
Yes. So yeah, moving along the molecule, the CF3 is in the right location as well. Yeah, very nice. The urea group here is where we made the linkage to the amino pyrimidine. So, and then we were ignoring the rest of the green molecule. So let me drop that off and bring back the pink molecule, because now this is where we're trying to link the two in. So this is not quite where I wanted it. So what we have here is, I was hoping that we could try and link to this amide better. I know this is a branch structure here. And so we have this pyrimidine, the aminopyrimidine. I was hoping to see it perhaps overlap a little bit more with this benzene ring here. And then the amide group up here is actually in pretty good place. There's going to be rotation here for the amide, so that could pick that up quite nicely. But I think what that's done, because it's not quite as close as I was hoping it could be, it means the rest of the molecule is a little bit offset here. So you can see here that looking at the nitrile groups, these nitrile groups are not quite as close as I wanted them to be. So a bit of so looking at the yellow structure, sorry, I think what we'd have to do is a bit more optimization around, or a bit more design around the aminopyrimidine, see what kind of group we could put there that could move this lower end of the molecule so it would overlap better with the pink purple structure that I was trying to get it to overlap with. So, but it's not bad as a first composite. So let's leave that there for the moment. Let's bring back the green molecule so we can have a look. And now we've got the yellow composite sitting in there. So that was the first docked structure. So I'm going to rate that pretty good, actually. I'm going to give that a 5-star ranking. I like that one. But there's 9 docked poses here. So that's pose number 1, pose number 2. Okay, so this is looking pretty good as well. Again, looking at the green up here, very nice. The yellow compared to green is very nicely structured. And again, you've got the fluoro with the trifluoromethyl overlapping, all looking very good. Bit more rotation here. It's moved the amide away from the original pink structure. And what I can see here is actually flipped the nitrile in the wrong direction compared to where I wanted. I wanted the nitrile here. I can see the nitrile over there. So not bad, but missing.
It does, though,
I'm going to give that 3-star.
take up more of the space like you were shooting for, but still pops it in the wrong angle for the nitrile.
Yeah, you're right. So it has rotated the aminopyrimidine structure, which has pushed this down further. I think if it could have flipped, this ring over, it would have put the nitrile closer to where I wanted it. So not bad. I like that one. I'll give that 3 stars because I'll give it 4 stars. The chloros, the nitrile is not quite where I wanted it, but okay. Pose number 3. Green, the overlap between the yellow and green is really good, but here you can see it's rotated the aminopyrimidine into the wrong direction. So I guess this is the amide down here. So that's not a good pose. It's moved this up in the wrong position. So I'll give that. I'm not even going to rank that one. For same kind of thing, it's moved the the triazole type structure with the nitrile in the wrong position. In this case, again, they all overlap very nicely up in this region. It's this region down here that's struggling with. Although in this case now, the nitrile is actually quite close. But in order for that nitrile to be in the right place, yeah, that looks really good. But now the ring structure is, it's still a bond away, but that whole ring structure is in the wrong position now. So I'll give that 2 stars. Pose 6, that's gone wrong somewhere. Yeah.
It tried. It was trying.
It tried, bless it. Yeah. We won't give that any stars. Pose 7. That's okay. With the top end again is okay. But again, I think the aminopyrimidine is rotated the wrong way, putting the nitrile in the wrong position. So not good. Yeah, that's not good. Pose 8. Everything seems to be misaligned to where we want it. And pose 9 has actually flipped it the other way around. So this ring should be up the other end. So I'm not going to rank that one either. But going back to pose number 1, I think that's as close as we can get with these, giving it the 5-star rating. And I think a bit of flexibility in that central section, or maybe even just a bit more movement in this region could allow it to fit fairly nicely. But not a bad composite. Not a bad composite. So the other thing we wanted to look at then was this lysine, right? The lysine 9. Was it 942, 941? Let's have a quick look. Select. Yep, 941. So we want to have a look at this with the TDU complex, right, where it moves that. Okay, so then this is the other structure with so the 8-TDU structure where the lysine 941 has moved out of the way. You can see here that now there's that gap there. So so we know that this residue can move in and out of that region there. So we know that was interacting previously. In this case now, it's going to be out of the way, probably not interacting, but we know that can go back in there. So I think this is not bad for a first composite of those 2 structures. But obviously some more work that could be done. I'm actually wondering if we took this pyrimidine and saturated that, made it give it a bit more flexibility and more freedom, different geometries, whether that might allow it to land and settle down a little bit better. but as a first structure, I don't know, pretty happy with it. I like the docking. I like the way it's docked. So be an interesting start for a composite lead op program.
Yeah, I definitely agree. It's fun to see you talk about 2 different molecules and then building, a chimera of those, and then watching it overlay really well. It was nice to see the vision come to life and watch it dock into the pocket. I'm wondering though, do you think so? I agree. Adding more saturation on this ring will then allow some more flexibility. Do you think an extra carbon or anything in there would be too much flexibility then? And you did, because you still have a little bit of area to work into.
Yeah, I've been thinking about that, like a methylene or something, putting a methylene in this position here. Could that give it, it would certainly give it a little bit more rotational freedom as well. And looking at all of these, it is about 1 bond away. So adding another bond in there, would that help? Possibly, yeah, which would allow it to stay as the amino pyrimidine, and we could put a methylene in place instead. Yes, yeah, be worth trying.
Yeah, I mean, lots of molecules, right? This is where you just start going crazy and building all the molecules you can think of.
A lot of molecules we can build off of here. I do like this region up there, and I do like the way that has, the yellow structure of ours has kind of split right between the branches of the pink structure. So I'm happy with that section here. I think, how do we, a lot of different things we could do on that lower section there. And that's not to even say that, that's just taking a composite of those two ligands and making the composite yellow structure. The other things I'm really interested in is just having a look at the potential for different hydrogen bond interactions around these different residues. We know the pi stacking should be in approximately the right place as well, but are there other residues that we could try and, pick up as well through hydrogen bond interactions and so on? So yeah, lots and lots of ideas that can start flowing from this.
Yeah, definitely. Well, I definitely appreciate the exercise of running through how someone in their everyday job using Nanome would be able to take crystal structures, move them into this three-dimensional space, devise a molecule or molecules that they would want to draw. We could then move those over to Mara, and then as a med chemist, you can
Yeah.
dock those your new
Yeah.
ideas in a protein, come back, evaluate, and then maybe hop in a meeting with your computational chemist and just be able to
Yeah.
get all of those ideas flowing faster than just, emailing back and forth.
Yes, for sure. Yeah, yeah. And just the richness of the structures that we can see here. Like I say, for me, being able to immerse myself into this protein and, keep the protein steady, move my head around to look at the different ligands, just creates a really much richer experience with seeing how the molecule is binding in there. And as you said, just the ability to quickly redesign compounds, redesign, do the docking, and start seeing what direction we might want to take the chemistry off in. And but yes, obviously maintaining communication with the computational chemist to make sure that I'm not going off on a tangent.
Yeah, which nothing wrong with tangents, but yes, it's nice to keep it all in mind.
Yeah.
All right, cool. Well, thank you very much. I appreciate all the time and working with you. This has been really fun.
Yeah, me too. Thank you very much. Thanks for the opportunity.